This is our model of a neuron. A neuron consists of a cell body with branchy fibers called dendrites. The dendrites will receive signals into the neuron. Some of these signals are excitatory, and they would speed the neuron up, and some of them are inhibitory, so they would slow the neuron down. Next, the myelin sheath, which insulates the axon of the neuron and helps to speed up impulses, so the axons. The end of the neuron has the terminal branches and terminal buttons. This is where the neurotransmitters will be released into the next neuron. The terminal buttons also contain vesicles that store other neurotransmitters that could be released for different reasons. This is the neuron at rest. A neuron fires an impulse when stimulated or sends signals. The impulse is a brief electrical charge that travels through the axon. This impulse is called the action potential. The level of stimulation needed to trigger the action potential is called the threshold. Neurons rely on chemical events to generate electricity. The charge at rest of a neuron is negative 70 millivolts. The resting potential of a neuron could have a fluid of negatively charged chlorine ions inside and positively charged sodium ions outside, with positively charged potassium leaking out. The axon surface is selectively permeable at rest, but when the neuron fires, the axon opens its gates and lets through positively charged sodium ions. This depolarizes that part of the axon, and it causes the next gate to open, and so on and so forth, and lets through positively charged sodium ions. This depolarizes that part of the axon and causes the next gate to open, and the process repeats itself. Meanwhile, the neuron pumps the positively charged sodium back outside so that it can fire again. In order for a neuron to fire at all, the number of excitatory neurotransmitters has to outnumber the number of inhibitory neurotransmitters. Otherwise, the neuron won't fire. When the action potential reaches the end of the axon on a neuron, it triggers the release of neurotransmitters. Neurotransmitters are chemical messengers that cross the synaptic gap and bind to receptor sites on the receiving neuron. The synaptic gap, also known as the synapse, is the junction between the axon tip of one neuron and the dendrite or cell body of another. The neurotransmitters will travel down the terminal branches and exit through the neuron. Tiny channels then unlock on the receiving neuron, which allows ions to enter the neuron, which will either excite or inhibit its ability to fire. Excess neurotransmitters will be reabsorbed into the receiving neuron in a process called uptake.